Hello everyone, today we are going to start pathology with the topic cell injury. So there are four parts of the cell which are vulnerable to the injury. These are rough endoplasmic reticulum, cell membrane, nucleus, mitochondria. What are the causes of cell injury? Most common cause is hypoxia, and hypoxia may be caused due to four causes like ischemia, anemia, cardiopulmonary failure, and carbon monoxide poisoning. Ischemia is caused whenever there is a decrease in the blood supply to any organ, and which can be caused due to atherosclerosis, thromboembolism, vasospasm. Other causes of hypoxia are infections, genetic disorders, hypersensitivity reactions, autoimmune diseases, physical agents like heat, cold, trauma, radiation, chemical agents, nutritional imbalances like deficiency of certain vitamins can cause hypoxia, or protein energy malnutrition, excess of vitamin A, DEC, that are fat soluble vitamins, or fats itself can cause hypoxia. And what are the outcomes of cell injury? Cell injury may cause irreversible cell injury or cell death. These are like necrosis can occur, necroptosis, apoptosis, or pyroptosis can occur. And reversible cell injury can also occur. In this case, hydropic degeneration, which is also known as cloudy swelling, can occur, or fatty degeneration can occur. Or the cell may sometimes adapt itself to escape the injury. So these are four types. Hypertrophy of the cells occur, atrophy, hyperplasia, or metaplasia can occur. And remember that dysplasia is not cellular adaptation, it is a pre-malignant condition. So this needs to be remembered. So, or intracellular accumulation and pathological calcification can occur as a part of cell injury. Likewise, proteins, fats, glycogen, pigments, lipofuscin, wear and tear pigment, and dystrophic calcification can occur, metastatic calcification can occur. The cellular aging can also occur. Now let's move on to the mechanism of cell injury, reversible mechanism. So how this mechanism occurs? Basically, there is a pump failure due to lack of ATP. So what leads to the pump failure? ATPase pump failure occurs, and efflux of potassium from the cell occurs, and sodium potassium influx into the cell occurs. So we all know that how sodium potassium ATPase pump works. So whenever there is failure of this pump, efflux of potassium and sodium and calcium influx into the cell, which will cause greater osmotic load, and when the concentration of the cell increases, water will enter into the cell and leads to the cellular swelling. Hydropic changes will occur, organelles will also swell up, and cell membrane blebs are formed. We can see in this figure, these are the blebs formed, and myelin figures are also formed. These are the myelin figures due to accumulation of water between membrane and the membrane phospholipids. Whenever there is accumulation in the membrane phospholipids, it will lead to the formation of blebs and myelin figures. So if ATP is absent, protein synthesis stops. First part was pump failure, which will lead to the stoppage of the second part is when ATP is absent, protein synthesis stops. Due to lack of ATP, there is no protein synthesis, increase in lipid synthesis and storage. Ribosomes dispersion of the ribosomes from the rough endoplasmic reticulum occurs, which we all know that ribosomes form proteins, so proteins will not form, and lipid synthesis will increase, which will lead to fatty changes. And there will be switch over to anaerobic glycolysis, because energy from aerobic mechanism will not be available. So glycogen in the plasma in the cytoplasm is consumed, and lactic acid is produced, and which will lead to decrease in the pH, because lactic acid is acid. So decrease in the pH will cause clumping of the nuclear chromatin. And what is the mechanism of irreversible cell injury? In irreversible cell injury, severe mitochondrial damage occurs. Mitochondria is damaged, so no ATP production by mitochondria, and large amorphous flocculant densities in the mitochondrial matrix are formed, which are composed of calcium and phospholipids. So this was the first mechanism, mitochondrial injury. Second mechanism is severe membrane damage. So membrane will be damaged, which will cause permeability of the plasma membrane to increase, and when there is increase in permeability, leakage of some important intercellular enzymes and proteins occurs, and also influx of calcium into the cell occurs. So what else will occur? Release of the lysosomal enzymes will occur. Whenever there is decrease in pH, increase in calcium in the cytosol due to increase in permeability, so this will lead to the lysosomal receptor irritation, and acid hydrolases are released. Lysosomal enzymes are released like RNase, DNase, proteinase, phospholipidases, which will break down all the components of the cell membrane like phospholipids, cytoskeletal proteins, RNA, DNA, and DNase will cause random breaks in the DNA, random breaks, and extract this DNA and do a gross gel electrophoresis. If we do electrophoresis, then smear pattern will be seen. Smear pattern. Now we will talk about nuclear changes. There will be pycnosis, that is condensation of chromatin. Pycnosis means condensation, and then karyoexis, which means fragmentation of the nucleus occurs. For initially condensation, then fragmentation, and finally dissolution of the nucleus, which means karyolysis. If we stain this with the help of hematoxylin and eosin, hematoxylin will stain nucleus, and eosin will stain cytoplasm and the extracellular connective tissue. We can remember like this: H is very similar to N, so hematoxylin will stain nucleus, and E is extracellular, so eosin will stain extracellular connective tissue. Now let's discuss about necrosis. Necrosis, cell death in the living tissue, is known as necrosis. Two processes that underlie the necrosis are: there are two mechanisms of necrosis. One is enzymatic digestion by the lysosomal enzymes, and second is denaturation of proteins. So first one is coagulative necrosis, due to denaturation of the structural proteins of the cells. Most common type of necrosis, coagulative necrosis, while liquefactive is seen only in the brain. And most common cause of liquefactive necrosis is hypoxia, except the brain, in which liquefactive is seen. Second cause is severe burns. First cause is hypoxia, second is severe burns, and third is dry gangrene. And Janker's degeneration, which means coagulative necrosis of the muscles like rectus abdominis. So Janker's degeneration is seen in the patient with a severe toxemia, example typhoid. And if we do hematoxylin eosin staining, cell outline is preserved, but cells do not have a nucleus in necrosis. This needs to be remembered. And increase in cytoplasmic eosinophilia occurs due to denaturation of proteins. Whenever the proteins are denatured, the cytoplasmic eosinophils, they are increased, and the loss of blue nucleus occurs on staining. And cytoplasm has a glassy appearance, Glassy appearance due to loss of glycogen, because glycogen is used as anaerobic glycolysis. So cytoplasm has a moth-eaten appearance. This is a question has been asked in various exams. Moth-eaten appearance due to loss of organelles, and most common organ where coagulative necrosis is seen is heart in myocardial infarction, most common. Others may be liver, kidney, spleen. Infarction. What is infarct? Area of necrosis, usually coagulative, produced due to ischemia. So infarct is basically necrosis, usually coagulative due to ischemia. Infarcts are maybe wedge shaped or triangular shaped. Wedge shaped are seen mainly in the kidneys. And two types of infarcts: wide infarcts and red infarcts. Wide infarcts are also known as pale or anemic infarcts, while red are also called as hemorrhagic. Wide infarcts can be seen in the solid organs with single blood supply, and red infarcts are seen in the spongy organs, like the organs which have dual blood supply. So wide infarct can be seen in heart, kidney, spleen, liver in hypovolemic shock, while red infarct in the lungs, where there is ample of blood supply, dual blood supply, liver. In liver both types are seen, but red infarct is more common. Organelles with collaterals, example intestine or the testes, ovaries. So in these also red infarcts are seen. Now let's move on to coagulative necrosis. Liquefactive necrosis. This is also known as colliquative necrosis due to enzymatic digestion of the cells by lysosomes. By lysosomal enzymes, dead tissue is converted into the bag of liquid. So, example brain hypoxia, always liquefactive necrosis. Abscess cavity, bacteria or fungal, cat gangrene. Okay. Now, what is caseous necrosis? It is seen in tuberculosis. Dead tissue has a white cheesy appearance and it is friable. Microscopically pink granuloma appearance is seen. Causes most common is tuberculosis, syphilis, the fungal infections can also cause it, like histoplasmosis or coccidomycosis. The fat necrosis can occur. So in fat necrosis, enzymatic fat necrosis or traumatic fat necrosis, these are the two types, enzymatic or traumatic. Enzymatic fat necrosis can be seen in patients with acute pancreatitis or abdominal cavity, mesentery, omentum or retroperitoneal fat. These are the sites where enzymatic fat necrosis can be seen, while traumatic fat necrosis can be seen in breast, subcutaneous tissue due to trauma. And mechanism of enzymatic fat necrosis is that the lipase released from pancreatic acinar cells attack the fat cell membrane. Lipase, okay. Fat cell membrane, free fatty acids are released from the fat cell membrane, and these free fatty acids combine with the calcium and cause chalky white deposits of the fat necrosis. Saponification of the fats occurs. Now let's study about fibrinoid necrosis, fifth type. The areas of the fibrinoid necrosis has a homogeneous pink appearance due to deposition of fibrin. As the name suggests, this can be seen on the hematoxin eosin staining. So causes of Fibrinoid necrosis are vasculitis, example pan, malignant hypertension, peptic ulcers, Escoff nodules of the rheumatic heart disease. These are the causes. You should remember by heart. And fibrinoid necrosis in the center is surrounded by the caterpillar cells. Okay. Aid. SLE, vasculitis, rheumatoid arthritis, rheumatic nodules contain fibrinoid necrosis. So fibrinoid necrosis is also present in these conditions. Now, what are the free radicals? These are the basic mechanism of injury, oxidative injury. These are the molecules with the unpaired electrons in the outermost orbit. Likewise, we can see that O2, there is unpaired electron in the outermost orbit, superoxide ion, and peroxide, the hydroxyl ion, which is the most powerful free radical. Chloride can produce HOCl, nitrogen peroxynitrite, free radical can be formed. And causes of the free radical generation are normal oxidation and reduction reaction occurring in the cells. Free radicals can be formed in the normal oxidation reduction reactions, or radiation can cause it due to hydrolysis, which can produce free radicals, or oxygen toxicity, infections, drugs, chemicals, reperfusion injury in the heart or brain. Free radicals damage cells in three ways. What are the three ways in which free radicals damage the cells? Lipid peroxidation of the membrane is the most important cause. Lipid peroxidation of the membrane. And DNA damage, oxidative type, oxidative damage to the proteins in the cytoplasm. So lipid peroxidation in the membranes, and DNA damage or oxidative damage to the proteins in the cytoplasm. These are the three ways by which free radicals damage the cells. And two reactions that generate free radicals are, one is Fenton's reaction. In this reaction, the hydrogen peroxide with the iron 2 positive forms hydroxyl ion, and this is the free radical, plus ferrous form is released. Second reaction is the Haber-Weiss reaction, in which the superoxide ion reacts with the hydrogen peroxide to form hydroxide ion. These enzymes that generate free radicals, these are the three enzymes that generate free radicals. One is NADPH oxidase, that is phagocytic oxidase enzyme or xanthine oxidase, and superoxide dismutase. All the things which are to be learnt should be noted very carefully. So there are two types of things. One is of learning, and another is conceptual. Conceptual things should be remembered and learnt very well. Superoxide dismutase generates and also inactivates free radicals. So this is the important point to be noted. Superoxide dismutase generates and also inactivates free radicals. Free radical removal mechanisms: antioxidants, which are very good for health, vitamin A, C, E, selenium, cysteine, glutathione containing compounds. These are very good for health, antioxidants. And enzymes like superoxide dismutase, which I have told that this also generates as well as destroys the free radicals. So it inactivates superoxides, SOD. And copper zinc, SOD, superoxide dismutase. These are found in the cytosol. And manganese, SOD, SOD2. SOD type 1 contains copper zinc superoxide dismutase. Type 2 is manganese containing superoxide dismutase, which is found in the mitochondria. So manganese, M for manganese, M for mitochondria, C for copper, C for cytosol. Okay, likewise we can remember it. So superoxide ion with the help of superoxide dismutase it will form hydrogen peroxide and water. So we can see that superoxide ion is converted into hydrogen peroxide, so it generates hydrogen peroxide. And catalase enzyme, which is found in the peroxisomes, it also inactivates hydrogen peroxide. So peroxisomes are organelles where free radicals are generated and inactivated. Next enzyme is glutathione peroxidase. Glutathione peroxidase is present in the cytoplasm and the mitochondria. It also inactivates hydrogen peroxide and hydroxide ion. Serum proteins. Albumin binds with iron and copper. Lactoferrin is iron binding. Transferrin is iron binding. And ceruloplasmin is copper binding. So free radicals can cause cell death by necrosis, apoptosis, and necroptosis. These are the three mechanisms by which free radicals can cause cell death. Brain is protected from free radical injury. Why? Because of the copper zinc superoxide, that is superoxide dismutase 1, superoxide dismutase. So whenever mutation of this enzyme occurs, amyotrophic lateral sclerosis of the brain occurs. Amyotrophic lateral sclerosis of brain. So most important ion involved in the cell injury is calcium. First ion involved is sodium. Most susceptible to the ischemic injury are neurons, 3 to 4 minutes. So neurons are very easily affected by ischemia, and followed by the cardiac tissues. Cardiac tissues can sustain up to 20 to 40 minutes, and tissue least susceptible to the ischemic injury are fibroblasts. Fibroblasts followed by the skeletal muscle. So first sign in all types of the cell injury except apoptosis, hydropic change. First sign we will see in any type of cell injury except apoptosis is the hydropic change. Hydropic change will occur in all the cell injuries except apoptosis. Okay. Then apoptosis is known as programmed cell death. It is an active process, needs ATP, and death of the single cell or single group of cells. No surrounding inflammation around the apoptotic cells. No inflammation occurs. This is active process, death of single cell, programmed cell death. Examples of apoptosis, there may be physiological apoptosis like embryogenesis, involution of the hormone dependent organs upon hormonal withdrawal, breakdown of the endometrium during menstruation, ovarian follicular atresia following menopause. Cell death is in rapidly progressing proliferating cells like the cells in the GIT, skin, respiratory tract. So apoptosis is very common physiological apoptosis. This occurs in our daily life. Death of the cells that have served their purpose. Example, death of the neutrophils at the end of the acute inflammation. We all know that in acute inflammation the neutrophils are recruited, so they die at the end of the acute inflammation. Elimination of the harmful self-reactive lymphocytes. So lymphocytes, when they become harmful, they need to be apoptized. And pathological apoptosis. Pathological apoptosis can occur in DNA damage, accumulation of the misfolded proteins, or viral infections like adenovirus HIV or viral hepatitis, which can lead to formation of concealment bodies. This should be remembered that viral hepatitis can lead to formation of concealment bodies. So these are apoptotic hepatocytes. Concealment bodies are apoptotic hepatocytes. Duct obstruction of the organ produces atrophy and apoptosis. Duct obstruction of the organs. So this can be seen in the salivary gland, pancreas, kidney, wherever there is ducts. Ducts are present, so there the obstruction during obstruction, apoptosis can occur in the salivary gland, pancreas, and kidneys. In kidney, ureter obstruction can occur. And what is the morphology? of apoptosis. Two enzymes can bring about apoptosis. These are caspase and endonucleases. So caspase break the framework of the cell. Cell will shrink and tight packing of the organelles will occur. What will endonuclease do? Endonucleases are the calcium magnesium dependent enzymes. So endonucleases break DNA at the specific sites. We all have studied in plus one, plus two. So internucleosomal regions. These are known as internucleosomal regions. So, so this is the whenever caspases occur, this it will cause the cell framework of the cell to destroy and cell will shrink. Tight packing of the organelles occur and 180 to 200 base pairs of the DNA fragments are produced after endonuclease acts on it. And on agarose gel electrophoresis, we will see step ladder pattern. Step ladder pattern is seen. Most characteristic feature is pycnosis, chromatin condensation. So I have already told you the steps in which initially there is pycnosis, then fragmentation, karyorrhexis, and then finally karyolysis occurs. So chromatin condensation, after chromatin condensation blebs are formed. You can see blebs here, and then blebs cut off from the cell and form apoptotic bodies. These are apoptotic bodies. Apoptotic bodies are nothing but after the condensation the blebs are formed, and after the blebs are cut off from the cell they form apoptotic bodies. So engulfment by the macrophages occurs. Apoptotic bodies are then engulfed by macrophages, and apoptotic bodies are removed because apoptotic bodies express thrombospondin 1 on their outer leaflet. Thrombospondin 1. So macrophages have receptor for the thrombospondin 2, which are absent in the normal cells. So they have to be removed due to this enzyme on their leaflets. Apoptotic bodies express phosphatidyl serine and phosphatidyl ethanolamine on their outer surface, and macrophages have receptors for the phosphatidyl serine and phosphatidyl ethanolamine. So macrophages are easily attracted towards these, and there is enzyme annexin 5 is a marker of the cells undergoing apoptosis. It is immunostatin that stains the phosphatidyl serine and phosphatidyl ethanolamine, which are expressed on the outer leaflet of the apoptotic body. So due to this we can stain. It is an immunostatin. Annexin 5 is an immunostatin that stains these, which are expressed on the outer leaflets of apoptotic bodies. Normal cells have phosphatidyl serine and phosphatidyl ethanolamine on the inner leaflet, while on the apoptotic bodies that I have told, they are present on the outer leaflet and thus remain unstained by the annexin 4. So the cells which are undergoing apoptosis will be stained by annexin 4 because the phosphatidyl serine and phosphatidyl ethanolamine, they are present on the outer leaflet, while in the normal, as I have told, that these are present on the inner leaflet, so they remains unstained by annexin 4. So microscopically, what will be the features that I have told, that the cell will be shrunken uh, because of the process of apoptosis. Hypersinophilic will be there, and chromatin condensation below the nuclear membrane. Pycnosis in nucleus. Pycnosis will occur. Chromatin condensation will occur. Rest we will do in our next video.